Welcome everyone, we're learning about lipid molecules in the plasma membrane. Now, if you extract lipids from the plasma membrane, if you extract lipids from the plasma membrane and put it into an aqueous solutions, then the lipid molecules can form one of the three structures in the aqueous solutions, that means in the water. The first structure is called Micelle. The second structure can form is liposome, and third structure is sheet-like bilayer. They can form one of the three structures. How it looks? A micelle is actually a monolayer of lipid molecules. Let's suppose the surrounding is aqueous solution, that means waters. There are water molecules present in the outside. So micelles are actually monolayer. Assume that all the surroundings are actually aqueous or water. So the lipids in the suspensions can form a monolayer. So it is actually monolayer of lipid, made up of monolayer of lipid. This is the structure of micelle. In the micelle, the lipids present are actually contain one fatty acid chain only. Micelle is actually monolayer in nature, so it is monolayer. And so if you take lipids in an aqueous solutions, that if that lipids contain a single fatty acid chain, then it can form micelles. In case of liposomes, two layers of lipids molecules are actually present. So in case of liposomes, two layers of lipid molecules are actually present, just like the plasma membrane. Now, this is the structure of liposome. This is bilayer in structure. So two layers of fatty acids actually surrounds the aqueous solutions. So here a lumen is present where aqueous solution can be present. Okay, so it actually surrounds the water molecule. And we know sheet-like bilayers looks like this. Two lipid bilayer. Now in case of micelles, the polar head group always face the outside. The polar head group of the lipids always face the outside because polar head group can make contact with water molecules. Here also the liposomes, the polar head group can face the outer side. But here, as the inside also contains another layer of lipid membrane, so the head group or the polar head group will be outside. And in case of liposomes, the inner side is actually lumen, called the lumen, and it is actually the aqueous solution of the liposome. So liposomes actually cover or actually surrounds the aqueous solution. So here aqueous solutions can be present. Also the aqueous solution present in the outside. And in case of sheet-like bilayer, also aqueous solutions are outside. Okay. Now the difference is here, in case of micelles, only one fatty acid chain can be accommodated, and in case of micelles, the inside don't contain any aqueous solutions because the inside is only contained with fatty acid chains, with one fatty acid chain. In case of liposomes, two fatty acid chain can be accommodated because they do not face the aqueous solutions. Only the polar head group face the aqueous solution. So there is an internal compartment and outside. Okay. Also here you can say, in case of sheet-like bilayers, the polar head group of the lipid molecule is outside that contact with the aqueous solutions, and the fatty acid chains are actually inside. Okay. So it will not face the aqueous solutions. Avoid the aqueous solutions. Now, what are the factors responsible for the formation of one of the three structures? The first factors, I can write here, factors. First factor is length of the fatty acid chains that are present in the lipid molecules. So length is important. Second point, you can remember degree of saturations. What is saturations? I have mentioned in my previous video that a fatty acid can have carbon-carbon double bond. If the fatty acid contains carbon-carbon double bond, then we call it unsaturated fatty acids. And if the fatty acid does not contain the carbon-carbon double bond, only carbon-carbon single bond, then we call it saturated fatty acids. Saturated fatty acids are actually straight. The structure of the saturated fatty acid is straight, but if there is any carbon-carbon double bond, the fatty acid look like this. So if there is any carbon-carbon double bond, the fatty acid actually have a bent or kink-like structure. Where there is a carbon-carbon double bond, let's suppose here is a carbon-carbon double bond, then there will be a kink. Okay, carbon-carbon double bond. So degree of saturation is important. Another important factor is that temperature. Now let's talk about micelles in more details. Micelles are actually rarely formed from the lipids present in the plasma membrane. So if you extract lipids from the plasma membrane, the lipids present in the plasma membrane in the aqueous solutions very rarely form micelles. And the reason is because of the presence of bulky fatty acids that cannot accommodate inside the micelles. Because I have mentioned that inside of the micelles there is no aqueous solutions like this. Here only fatty acid chains are actually present. Actually, they actually exclude the water molecules. They actually simply aggregate and form micelles. So rarely formed from the lipids present in the plasma membrane because of the too bulky fatty acid chains present in the plasma membrane lipid molecules. But you can make the micelles with the lipids that are present in the plasma membrane with the help of one enzyme called phospholipase. Now, how this enzyme can create micelles with the same lipid molecules, let me explain. In my previous video, I have mentioned that a phosphoglyceride is one major lipid molecule present in the plasma membrane. And the structure of phosphoglyceride is like this. The phosphoglyceride contain a glycerol, that is the backbone of the molecule. And this glycerol is actually 3 carbon molecule. First carbon, second carbon and third carbon. With the first carbon, one fatty acid chain is attached. With the second carbon, another fatty acid chain is attached. With the third carbon, a phosphate group is attached. Okay, a phosphate group is attached. That's why it is called phosphoglycerides, because it has a phosphate group and glycerol. Now, the head group can vary in case of phosphoglycerides. I have mentioned the head group could be another glycerol, it could be a choline, ethanolamine, serine or inositol. So this is the basic structure of phosphoglycerides. So I can write here an example of phosphoglycerides. Now, phospholipase enzyme can have actually there are 4 phospholipase enzymes. One is called PL, that means phospholipase A1. This enzyme can cleave the bond between the first carbon and the fatty acids. So it can cleave here to remove the fatty acids. There is another phospholipase enzyme is present, that is phospholipase A2, that can cleave the bond between the second carbon and the fatty acids. So it can cleave here and it can remove the fatty acids. And there is other 2 phospholipase that is called PLC, that can cleave the bond between third carbon and the phosphate. So it will remove the phosphate group, and with the phosphate group the head group can be removed. And there is another phospholipase enzyme called phospholipase D that can cleave the bond between the phosphate and the head group. So there are 4 phospholipases that can actually cleave the phosphoglycerides in different positions. Now, if you treat with any of the 2 phospholipases like PLA1 or PLA2, you can remove the fatty acids from this structure. See, if you, let's suppose if I treat this structure only with PLA2, then this structure will be look like this. It will not have the second fatty acids. If you cut with PLA1, then the first fatty acid will not remain, so it will have only a single fatty acid. And as I have mentioned, in the micelle, the lipid can accommodate only as one fatty acid chain. So by the help of phospholipase, if you treat phospholipase with phosphoglyceride, then only it can, especially PLA1 or PLA2, not with PLC and PLD, because it will not cleave the fatty acid chain. So then it can, this structure, this type of lipids, this type of lipid is called lysophospholipids. Remember the name. When we treat with this type of enzyme PLA2, the structure is now actually different. At first phosphoglycerides contain two fatty acids, now it has only one fatty acid. This type of structure is called lysophospholipids. Lysophospholipids. Okay, see, now lysophospholipids can accommodate in the micelles and can form the micelles. So this is all about micelle structure. Now let's talk about liposomes. Liposomes are actually very similar like the plasma membrane that surrounds a cell. Okay, what happens basically when you actually take the extract of the lipid molecules that are present in the plasma membrane in an aqueous solution, these lipid molecules actually start to aggregate. Okay, they exclude the aqueous molecules, that means water. They try to accommodate with their own molecules. That's what do I mean? That let's suppose this is the polar head group of a fatty acids, and these are the two fatty acid chains of the lipid molecules. Now what will happen, I have mentioned previously that the fatty acid chain is actually hydrophobic in nature. Hydrophobic in nature. And this polar head group, basically this phosphate and the head group makes the polar head group. So this is actually hydrophilic in nature. This is polar head group. Polar head. So this makes this polar head group is actually hydrophilic in nature. That's why when I have mentioned that those molecule or structure contain both the hydrophilic and hydrophobic, we call this type of molecule amphiphilic. Amphiphilic or amphipathic, whether you can remember, amphipathic. Now what will happen when you extract lipids from the plasma membrane and keep it in an aqueous solutions? That means the lipids are actually surrounded by water molecules. There are water molecules. Now as much as lipid molecules will be present, they try to aggregate with each other to avoid the water molecules, because this is hydrophobic. Now what will happen, number of lipid molecules will come close to each other, and actually the fatty acids will remain here. Only the polar group that is hydrophilic in nature, they will make contact with the aqueous solutions with ionic or you could say hydrogen bond with the water molecules. So this polar head group can contact with each other also, with ionic or hydrogen bonds, also with the water molecules. Now if there is any edge in the membrane, let's suppose if there is any edge that is exposed to the water, that will create a problem, because these lipid molecules or the fatty acids actually avoid water. They are actually non-polar in nature. We call this hydrophobic nature is called non-polar. I have already discussed, and we know that water is actually polar molecules. They are polar molecules. That's why I have written here polar head group that can interact with the polar molecules like water and water soluble solids. So these fatty acids are non-polar, and we know that polar and non-polar do not mix. Water and oil do not mix. Water, fat do not mix. So they try to avoid the water molecules. And let's see if there is any edge. If there is any edge in the membrane, that will interact with water, and that is energetically unfavorable. That's why they actually close in on itself. So this fat or these lipids close on itself. That means it has a self-sealing property. Remember the term, it has, the lipids has a self-sealing ability or property. Now, if you take a sheet like bilayer in an aqueous solutions, then what will happen? The edge of this sheet of bilayer interacts with the water molecules, and that is energetically unfavorable because fats are actually non-polar in nature and waters are polar in nature. So both water and fats actually avoid each other. That's why the bilayer, sheet like bilayer, has a tendency, or it has a always tendency to close on in on itself. That means it has a self-sealing property, and it will make a spherical structure just like the liposome. So this is fundamental to the creation of cells. This is one important fact we have to remember about the liposomes. Okay. Now, this artificial bilayer has three important properties we have to remember. So I have to remove this writing. Okay. This artificial bilayer has three important properties. Remember the property. The first is they are virtually impermeable to all water soluble solutes. That means water soluble solutes means polar in nature. Those molecules are polar in nature, like glucose, fructose, or it could be ions, or it could be amino acids, or ATP, or nucleic acids. Those are polar in nature. That means water soluble. That solutes cannot enter through the they cannot enter through the artificial lipid bilayer. It has been found. The second point we can remember that this structure, this liposome structure, is very stable. They are very stable. The pH or the ionic contents of the aqueous solution can vary huge, but the architecture of this structure, this bilayer structure, remains stable. So it's very stable. And third point that I have already discussed, that there will be no edge. They will not actually tolerate edge. So no membranes can have a edge because they will always make us shield compartments. So they make sealed compartments. So that's why here could be aqueous solution, outer side could be aqueous solution, but they will seal it. So it makes a sealed compartments, and that is the fundamental to the creation of cell. So this is very important. Now I will talk about the usefulness of liposomes in research field. So let me remove also these writings. First is liposomes helps in membrane research. Okay, because you can create artificially this kind of structure. Second is this liposome can act as a drug delivery vehicle. So it acts as a vehicle to deliver drug. Now, if the drug is actually polar in nature, that means aqueous solution or water soluble, then you can keep this drug here. You can keep this drug. Let's suppose this is the drug. You can keep this drug here. But if, let's suppose, if the drug is actually fat soluble, then you can keep the drug here. So you can deliver the drug with the liposomes. Now, if you want to target a particular type of cells, let's suppose any particular population of cells are actually performing some wrong functions, then if you want to target those cells, okay, so you can actually tag these liposomes with antibody. Let's suppose this antibody. Now we know that antibody can specifically bind with an antigen. Let's suppose that antigen is expressed by the particular disease causing cells, then this liposomes could be targeted very specifically, non-randomly. So these liposomes can be targeted very specifically can be targeted by the use of antibody. Okay. Next, we it has been found that the previous clinical studies with liposomes as a drug delivery system actually made failure, because as soon as these liposomes enter inside the body or system, the phagocytic cells in the immune system actually engulf it and clears from the system. To overcome this obstacle, scientists actually developed one category of liposomes called stealth liposomes. They have made one category of liposomes that is called stealth liposomes. These liposomes are actually coated with a synthetic outside. The outside is coated with a synthetic polymer. One example of this polymer is polyethylene glycol. This is used in laboratory, polyethylene glycol, or in short we call PEG. Okay, so stealth liposome, one example of stealth liposome that is actually used in the therapy is called Silix. This stealth liposomes carry one chemotherapeutic drug, that is doxorubicin. This Silix or these liposomes actually acts as a therapy for metastatic breast cancer, and it is actually approved for the therapy Silix. So liposomes can be used differently for membrane research and for drug delivery also. Okay, now this is all about the liposomes, and I have mentioned that liposomes, I want to mention that liposomes is actually bilayer. Remember this is bilayer. Okay, now how could you make sheet-like bilayer? Because I have mentioned that if you keep these lipid molecules in the aqueous solution, they actually immediately seal itself to avoid the interaction with the water molecules. So how could you actually make a sheet-like bilayer? So you can make also this. Let's suppose this is a chamber where aqueous solutions are present. That means waters are present. Let's suppose this is the water. Now if you divide these aqueous solutions with glass, let's suppose we have separated these solutions with a glass, and now if you can put a small gap in the glass, and now if you put these lipids, then the lipids will form a shield like this. It will seal the gap, and the fatty acids will be here. So I cannot draw actually exactly how it will look, because it will actually comes together and will not form a spherical, because they are sealing itself. So then you can get this type of sheet-like bilayer. But if you just put these lipids in the aqueous solution, it will immediately form the liposomes. This is for sure. So this is sheet-like structure. Let me see if I have already covered all the informations. Okay, one thing I think I have forgot to mention, that these interactions with the lipid molecules in all these cases, like here or here, they actually interact with van der Waals interactions. And I have mentioned that the polar head groups interacts with each other and with the water molecules. Here I have mentioned with ionic or hydrogen bonds.